This service allows you to import your own expression data, regardless of how you have generated it, whether it's generated using RNA-seq or microarray, as long as your data is in differential expression format, which is basically as log2 fold change. It will allow you to upload your data as a simple tab-limited file or the Excel file, where the rows are your genes, and you need to pick the gene identifiers which can be understood by the system. So it can be ref-seq locus tag. If you have your own genome, you can use the BRC identifiers, which is like fig, bar, and then some numeric identifier that we have. So you can use those IDs as your first column, right? Your rows are, each row is a separate gene, and then each column can be one of your experimental condition. So it can be the example that Clark showed you earlier, where you had 3 differential expression condition: cholesterol versus meropenem, cholesterol versus control, meropenem versus control. Those 3 can be your columns. And then each of the value is going to be the log2 fold change, indicating the differential expression of a given gene in a given pairwise comparison or the contrast that you had. So that is the data. And you can pretty much fake any experimental data that you have as long as it meets that nature, or if your values are of that nature, it doesn't matter. You can have differential, let's say, protein abundance data also. So if you are doing proteomic experiment using mass spec or something, and if your data is basically the abundance of a given protein in condition A versus condition B, you can use that data also. And as long as you have follow the data format, you can use it, you can upload it. And then what this tool is going to do is going to import your data in the heat map that Clark just showed you. So once you import your data, you can do all the filtering based on include, exclude, don't care, load the heat map, do the clustering, and then do the keyword filtering and all those things. So we have one data set available that I just loaded in the common workspace. So if you go back to the BV-BRC workshop folder, and if you go to the expression import here, this is the data. So there is a short README file over here, if we can show it quickly. So the data comes from this particular publication that's mentioned over here. It's a famous publication from Sangudekar, and the data is about Escherichia coli MG1655. So what they have done here is basically they have knocked out each of the non-essential, if I remember correctly, or this is the expression across. Yeah, so this data set was generated using microarray, and this is one of the most comprehensive gene expression data set that was available at the time. But for Escherichia coli, it was treated with about 250 different experimental conditions. It includes like temperature and pH and various antibiotic compounds and different growth media and things like that. And then they're trying to assess the expression of various genes across all of these conditions. And typically, like the reason that this kind of large-scale experiments were run early on is basically try to understand the function of the uncharacterized genes. So you have a lot of proteins and genes which are labeled as hypothetical. You have no idea what they are doing, not at least using the typical bioinformatic tools and similarity-based methods and things like that. So the goal of this experiment were that, let's say, if you find out that certain genes were highly up or down regulated under certain experimental condition, maybe you can generate some hypothesis about the function of those genes that maybe participate in the stress response or pH response or the temperature difference and things like that. And then you can run further experiment to actually characterize the function of the gene. So that's the data set. And if you go further down, you might see more information about the growth condition. These are the kind of heat maps that are presented in the manuscript. And it's a very rich data set. So what we did is we downloaded that entire data set. So that's the data set, by the way. And if you go one level back, this is the actual data file that we are trying to upload. So all of that data is a text file, but you can download it and you can open it in Excel also. And this is the data, right? So as I said, this B001, 002, those things are the ref-seq or gene bank gene identifiers. And then each of the condition here, you can see those are the experimental conditions. So what it's telling you basically is that the treatment here is 0.2 molar sodium azide solution. And then your sample or the reading is taken at 5 minutes after the exposure or 10 minutes after the exposure and things like that. Those are the experimental conditions. And there are about 250 different columns like that. So if you have your own experiment, or if you are interested in reusing published gene expression data set by somebody else, what you need to do, find the publication, go to supplemental information, download their gene expression data file. You may have to massage it slightly to make sure that it follows certain format, get rid of the extra columns that they may have about the gene annotations and things like that, and then just convert it into slightly simpler format like this. And then once you have it, you can go to the tools and services, expression import. So the question is that if, let's say, you are doing your experiment and you want to combine your data with somebody else's data, some previously published data, can you combine the two data sets and then load it as a single file? The answer is yes. You may have to do some juggling or massaging of the data to make sure that your data and the other data aligns. So some of the problems can be, are you both using exactly the same organism or the genome in your experiment, right? Are you both using the same gene identifiers? So that would be another thing. And then once you have it, you can map the data and you can load it. If your data, you need to look at the measurement units, also the values that you have in there. If the nature of the data is, let's say, differential expression and you both are using, let's say, log2 fold change or something, then yes, you can combine the data. So the only value that are currently understood in this expression import tool are the log2 fold change. It's assuming that your values are of that nature. In future, when we have extended support for RNA-seq data, we can probably also modify this to accept TPM values or something like that, but that's not supported right now. The other thing you'll have to keep in mind is that many times when people publish their data, they have already applied some kind of filters or threshold to throw out non-significant genes. So if you have already applied, let's say, when you're publishing your data, you are reporting only the significant genes based on the p-values or based on log ratio of 2 or higher or something like that, then you are comparing your RNA-seq data with only the subset of data from their experiment. So you'll have to keep that into mind when you are combining two data sets and loading it into heat map or something to keep in mind. Right now, it doesn't allow you to select two files. You'll have to do it outside of the system. So you'll have to somehow merge those two data sets into a single tab limited or Excel file before you load it here. It's going to be a bit complicated, but that's something that we can maybe try to support going forward. Okay, so I'm going to go to that public workspace here. Then you'll have to find the BV-BRC workshop, expression import, and then select the Sangodekar microarray data all conditions. Click OK. And then there are some metadata or additional information that you can provide over here. It doesn't affect the outcome or how the data is displayed. One of the information here is whether it's transcriptomic or proteomic experiment. And there is no difference in how the data is treated or portrayed at this point. So it's an optional thing. Whether your genome type is bacteria or archaea, I think that, or the eukaryotic host. Whether your ID type is the BRC ID, Patrick feature ID, or RAFSeq locus tag. So in this case, the ID type that we have is the RAFSeq locus tag. And then what is my target genome? So in this case, the target genome is MG1655. So that's the E. coli, Escherichia coli MG1655 strain. And as you can see here, this is another problem that we have these days, that the same strain is sequenced by pretty much anybody and everybody in the whole world. There was a time when MG1655 was a reference genome, and there was only one genome of MG1655. Now there are probably 100 genomes with the same strain name. So the one that we are interested in here is the classic one, or the one that was published the first. So you can probably get rid of all public and my genomes if you want, and that will restrict your choice to only or the genome that we actually are interested in. And you will see that there is a bracket and reference label in front of the genome name. So that's the one I want to actually select. And then you can provide what's your experiment. So in this case, let's say E. coli. You can put whatever name you want. Description, you can provide it or you can leave it blank. I think it doesn't really matter. I'm going to copy the same thing here. Organism name, this is MG1655. So this is just you are providing the taxonomy name. PubMed ID, if you have it, you can provide it. It just helps link your data or provide some pronouns. And then where you want to put your data. So I'm just going to leave it in the default folder that's specified here, which is the experiments. And there is an optional metadata file that you can also provide, and it needs to be in a specific format. So the first column that you have needs to match the sample identifiers or the sample names that you have provided as the column headings in your data file. And then all the other columns, you can provide information about the time point, the treatment that you have used, and maybe whether it requires any gene knockout or gene mutations or not, and things like that. So it just makes your data more rich and more informative, but it's optional. And then once you have provided this information, you can just click submit. It should take probably just few minutes, maybe about 5 minutes or 10 minutes at the most to map the data. But if you don't want to wait, we can go back to the workspaces, the workshop folder that we have there, then go to expression import, and then we can look at the job that I have copied over here. So this is a completed job done in exactly the same way. And then if you click on that, none of these files are going to be important or of any meaning to you. These are just the internal data structures that are generated to feed the interactive gene viewer and the heat map viewer that we have. But as far as you are concerned, for most users, it's not going to be useful to you. So the thing that you are interested in here is basically going directly to this view button or going to the experiment. So this gives you summary of your data. So this is some of the information that I provided when I submitted the experiment. So it's telling me what the organism is and how many of the genes in my data set are actually mapped to the version of annotations that we have in the database. So remember that every genome that we have in BRC, it has gone through our own annotation pipeline and it produces its own set of annotations, right? So that is different than the version of annotation that you see at GenBank or RefSeq. So in this case, what it's telling me that out of 3,600 genes that were there in my data file, 3,450 of them were able to map to the BRC annotation. So the question is, why there is this difference? What happened to the remaining 150 genes? And the answer there is that most of the annotation systems, they'll do a reasonably good job of annotating well-characterized genes, the genes that have function, genes that belong to metabolic pathways and things like that. But then in a typical bacterial genome, you have about 30 to 40% of the genes, which are hypothetical genes. We don't know anything about the function of those genes. In that also, there are probably going to be about 5 to 10% of the genes, which are really small in their length, right? So these are the genes which are typically anywhere from 30 to 70 or 80 amino acid long. And a lot of these genes overlap with other well-characterized genes or overlap, two hypothetical genes overlapping with each other. And most of the annotation systems, they have some kind of heuristic in their algorithm to say, when you encounter this situation, when there is a significant overlap of two genes, how you are going to resolve the overlap, how you are going to decide which one to keep and which one to throw out, right? Because you don't want to have too many of these genes which are overlapping with each other. It doesn't happen that often in bacteria. So typically what they'll do is they'll run some kind of check against maybe BLAST databases or something, and the gene that has less evidence in terms of sequence similarity or something, those genes will get tossed out. The differences that you'll see like this are those small hypothetical genes where the version that's in GenBank and RefSeq and the one in BV-BRC, two systems decided differently which gene to keep and which gene to throw out, right? So in most cases, if you look at what genes are not included here, they are all going to be uninformative, and it's not going to probably change the analysis that you are doing for your gene expression data or the outcome that you are going to report in your publication. So having said that, remember the additional metadata file that I mentioned earlier? If I had uploaded that additional metadata file saying strain is there, what kind of gene modifications are used, what is the experimental condition, time point, and those things, then this table would have been better populated. You also have summary of out of total number of genes that you have here, how many genes are significant in terms of the log ratio threshold. So the default threshold that we are applying here is log ratio of 1. So anything that has the fold change is between -1 and +1, we are considering them as insignificant. It just gives you some idea of how many genes are interesting in this particular data set. And the Z-score is computed based on the expression of all the genes in a given sample. Imagine all the genes following a normal distribution in a particular sample. Most of the genes are going to be somewhere between -1 and +1 or in that mid-range. They are not significantly up or down regulated, but then you are going to have at the end of that bell curve, some genes which are going to be significantly up or down depending on the experimental condition that you have subjected it to, right? The z-score threshold here, it's specifying how many genes are there which are more than 2 standard deviation away from your sample mean. So these are the top 5% genes that are either downregulated or upregulated. And you can play with this and you can actually get the same data when you go to the heat map. Now, the reason we wanted to go do this whole expression import is basically to get to this view. So now from that tab limited file that we had, the one that we downloaded from a supplemental information in that publication, now I have all of that data. So imagine you have 250 conditions here. I think that's about right. And 3,500 genes, right? So that's the total amount of data points that you have in your data set here, 3,500 times 250. And anytime anybody publishes the result, there are going to be some genes which are going to be identified as significant. They are going to comment something about their pathways, something about what conditions they are up and down regulated in. But there is always this question that there is a huge amount of data that is generated as part of this experiment. And only a fraction of that is reported in the analysis result that's reported in a particular publication, right? But if you are interested in any other E. coli gene, you should still be able to use that data to ask question in terms of the gene that you are interested in or the pathway you are interested in. And that's what this tool allows you to do. Once you have imported the data, now you can take your pathway or the genes that you are interested in. You can say, hey, what's the expression of this gene in this particular data set? So that you can maybe learn something about your own genes. So it allows you to reuse somebody else's data in a meaningful way here. But once you have this, you can apply the same controls over here. So I can say log ratio threshold of 2. I'm just being kind of like too stringent here, but just to reduce the amount of data. Z-score is also, let's say, to filter this. It still gives me about 2,000 genes. And then I can go to heat map. And I can look at all of that data. So now you have 2,000 genes and about 250 different conditions, okay? And you can use all the filters just like we talked about earlier. So you can say filter for transporters or the antimicrobial resistant genes or something like that. You can also apply cluster. I think this might be too much data to cluster because it has some size limit over here. So there's so much of data, it's probably not going to work. But you can also say, give me everything that's upregulated in, let's say, 1 or more conditions here. And then, of course, that's much smaller set here, and then you can apply clustering if you want. So I'm not going to go into too much detail, or I don't have any interesting data points to highlight over here, but that just gives you some idea of what you can do with this. You can also select things from here, and then you can follow up on that. So that's another advantage. Once you have identified up and down regulated genes from here, you can either download the data, or you can say, show these genes or add this set of genes to save it as a group. You can go from experiment data to say, okay, all the genes identified or other upregulated in condition A, and then you can save it as a group. That's a good way of creating your gene sets and doing the comparison later on. We also have a tool which allows you to do a set comparison across multiple groups. So let's see if I can show that quickly here. So I'm going back to my table here. So let's say I'm interested in maybe too stringent. Okay. Just only one gene. I want something that's more significant. All right, 73 results. So what is this condition? This is recovery after NAP buffer, whatever that is. I have no clue. But I can take this and I can say save all of these features as a group. So this is my new group, and then I can say feature group folder, and then experiment NAP. And then I can go to the second one here, and I can, or maybe this one, I guess. All right, so this one is 58 genes. So I'm just going to select this, save this as my second group. And this one is LB plus glucose, I believe. So just take this, create a new group, feature group, and then this is my experiment glucose. I don't even know the exact condition, but I'm just going to just use some dummy name here and then add. So now the question can be, let's say, okay, what's common between these two? And You can ask this question for any two feature groups you have created. Let's say you selected some things which are conserved in using your protein family heat map. You found something interesting from proteome comparison. If you have multiple feature groups or genome groups, how do you find out what's common between the two groups, or what's unique between them? So we have one tool over here. So you can go to your workspaces if you have saved any feature groups or something. If not, then you can just see this quickly. So I can select two groups over here, and then it's going to give me this option of Venn diagram tool. Okay, and if I click on that, it is going to give me this diagram here, which says, here is my group 1, here is my group 2. This one has 58 members, this one has 73 members. And then there are 16 features or proteins that are common between the two, right? And if I'm interested in kind of knowing what these 16 are, I can select that and I can create a new group. So these are all the features that are common between the two. And you can see them here. And if I'm interested in saving this, I can create a new group from this selected region. So now I'm taking intersection of group A and group B, and then creating a new group out of it. It is automatically selecting a group name for me. It's not allowing me to select my own name. But if I go back to my folder that I had, here is the new group that just got created. It does give me meaningful names. So it's basically set 1 for the group 1, group 2, and end is your intersection between the two, right? So if I had selected, let's say, either left or right region of the Venn diagram, then it would have come up with a name which says present in A but not B or something like that. And then if I want to see what these features are, I can look at their names and functions and all those things over here. But you can imagine scenario where, let's say, you have multiple conditions. You can say, give me all the genes which are upregulated in colistin versus meropenem. I'm going back to the antibiotic resistant example that Clark was showing earlier, right? And save that as a group. You can say, okay, give me all the genes that are upregulated in colistin versus, let's say, control. You can save that as maybe another group. And you can also have, for example, a set of genes that you identified from certain pathways, glycolysis pathway or something else. And you can save those genes as feature groups. And once you have saved that as feature group, so for example, now if I go back to Escherichia coli over here, I'm going to the genome just to extend the example that I was just showing you. This is the reference genome that we used earlier. Okay, so I'm going to specialty gene. I don't know whether we talk much about specialty gene or not. But specialty genes are basically the genes which are like potential virulence factors, antimicrobial resistant genes, human homologs, and potential drug targets. So these are all generated as part of the annotation pipeline. I think Jim talked about it yesterday. But I can go over here, and if this page loads, so I'm going to select all the antimicrobial resistant genes from here, for example. And I'm going to select all of this and save this as a feature group. So scroll further down, save this, create a new group, and I'm going to call it E. coli AMR genes, add. And while we are here, I'm just going to create another group here, which is instead of antimicrobial resistant genes, let's say the virulence factors. Or maybe, okay, let's try this and see if that works. And save this as a separate group, call it E. coli virulence factor genes. So I can go to my groups now. I added them all to my feature groups folder. So now I can select, let's say, my AMR genes and the genes that came from my gene expression data sets. Remember that the two other groups that we created earlier. So this is gene expression with the Na phosphate or whatever that condition was, and then gene expression in the glucose condition. All the genes were upregulated. And then I can select those two and create a Venn diagram of those three things. So interestingly, there is nothing common in all those three sets. Or I can go back to the same one here and I can say, okay, are there any genes that are common or labeled as both the virulence factor and the AMR genes, right? So I can go here and generate a Venn diagram for that. So the point I'm trying to make here is that when you are working on an analysis project, typically what you'll do is you'll do some search to find the genes or genomes of interest. Then you'll include them in your proteome comparison analysis, protein family analysis, or pathway analysis or something. You'll come up with a set of genes which are going to be somewhat interesting. And then every step of the way, think about creation of group as you are creating these checkpoints, right? Every time you find something, you save it in your workspace so that next time you come back, you won't have to go through all those analysis steps again. You have that set for you. And then as you keep on saving this set, you can later on use to say, hey, what's common between these two? What's unique between these two? And then you can ask questions like that later on.